My name is Thomas John Martin, known as Jack, and I'm Emeritus Professor, University of Melbourne, and I was Director of the St Vincent's Institute of Medical Research until just over 20 years ago, and I now am here on a retired basis. I just come in every now and then and pay attention to what my former colleagues continue to do. Well, I suppose the first thing that influenced me to turn to an interest in bone was when, a few years after graduating in medicine, I'd just completed a physician's qualification. I went to London and I met up with Ian McIntyre, who just discovered calcitonin, a hormone which influenced bone. And I was able to work with him on it, and we produced some things together. And it gave me an interest in bone that led to all the other things that I followed up with over the next years. And then I came back to Melbourne, and I was again fortunate to work with a mentor named Roger Millick, who had done a lot of work on the other main hormone of bone, the parathyroid hormone. And I worked with him for several years, during which I became interested in the cells of bone. And at that stage, it wasn't really known clearly that bone had anything as complicated as cells in it, but we started to be interested in those. And they were two of the main things that happened to me in the early years. And then I subsequently developed methods of looking in detail at the cells of bone, what they did, how they communicated with each other. And that has occupied my life for much of the last 40 or 50 years. At a stage when it was not really possible to isolate cells of bone, there was a bone tumour, a cancer of bone which we knew about, called the osteogenic sarcoma, the osteosarcoma. It was a tumour of cells that formed bone. And there had just been developed a way of inducing that tumour in the rat by giving repeated injections of radioactive phosphorus. So I did that while I was in Melbourne, working at the Royal Melbourne Hospital first, and then at the Austin Hospital. And I induced this osteogenic sarcoma in the rat and was able to isolate the cells from it. And these cells made a substance like bone, and they were very responsive to the hormone, parathyroid hormone. So I used those in the study of the actions of parathyroid hormone and used them for many years. And from that, we developed what we call a clonal line of those cells, which we made available eventually all around the world, and it's been used in hundreds of studies around the world and publications since that time, since the 70s and 80s. And one of the things that I noticed about that was that the ways in which parathyroid hormone acted upon it were very similar to what you'd expect of how parathyroid hormone would act upon the cells that formed bone, the so-called osteoblasts. And the osteoblasts were one of the three main cells of bone. The other cells of bone, the osteoclasts, continually break bone down, and they were thought to have entirely different origins, the osteoblast and the osteoclast. But what we found was that the osteoblasts, beginning with the osteosarcoma cells, but then with the osteoblasts that were soon able to grow from bone, had a responsiveness to parathyroid hormone and to other substances that very closely resembled the responses of bone breakdown to those substances. And so I developed with a colleague in the United States, Gideon Rodan, who's since died, that there was a communication between the bone-forming cells, the osteoblasts, and the bone breakdown cells, the osteoclasts, and that when certain substances, notably parathyroid hormone, acted upon the osteoblasts, that activities were generated that promoted the formation and activity of osteoclasts. And that was a completely new hypothesis. It created a lot of interest and a lot of opposition at first, but we and others developed methods of showing that it was pretty well true that the formation activity of osteoclasts were governed by activities that were generated from cells of the osteoblast lineage. And this proved to be a very valuable hypothesis because we generated it in the early 1980s, and over the next 15 years, many efforts were made, including by us, to find what it is that the cells of the osteoblast lineage produced that generated the activity of osteoclasts. And eventually, by the late 1980s, we weren't successful in finding what the substance was, but a couple of pharmaceutical companies did. And it was a substance called rank ligand, which was made by the osteoblast and which acted to promote osteoclast formation. And if you inhibited this rank ligand, you stopped the breakdown of bone. And that indeed was the basis of the development subsequently, since the late 1990s, of a drug which is an antibody to rank ligand, which prevented the breakdown of bone and which has been used in the treatment of osteoporosis for that reason. Osteoporosis, the bone loss that's common particularly amongst elderly or women post-menopause, is due to the loss of bone, and it's predominantly due to the overproduction and overactivity of these cells that break bone down, the osteoclasts. Well, this drug, which was based on the hypothesis that we developed, was established then and is still the most marketed drug for the treatment of osteoporosis in the world. It's called denosumab. It's sold in Australia as Prolia. So that was one major aspect of our work. But in the course of those years, when we were trying to work out how the osteoblast controlled osteoclast function, we became very interested and produced a lot of work which was fairly fundamental work on how the osteoblasts change during development and how the osteoclasts change during development and how the cells of bone communicate with each other. And communication between the cells of bone is very important, and it takes place not only by this rank ligand that I mentioned, but also by other short-acting proteins, which we call cytokines, which are produced by virtually all cells and which mediate the communication between the cells of bone. That had an effect on osteoporosis, but over those years, when I came back to Melbourne from where I went to work in Sheffield, I came back and worked at the Heidelberg Repatriation Hospital, which was where we looked after old soldiers of the First and Second World War. And very common amongst those soldiers was lung cancer. Many of them were smokers. And that lung cancer was often associated with a high blood calcium, the calcium having been leached out of bones. And I developed quite an interest in this complication of lung cancer, this high blood calcium, and I worked on it for quite some years. And for quite a long time it was thought that those cancers inappropriately produced parathyroid hormone. And so we worked on what that substance was. And eventually I found out that a cancer that we'd worked on in Sheffield before I came back to Melbourne produced an activity which resembled very closely what we would expect of such an activity from lung cancers. And it was very like the activity of parathyroid hormone. So we set about trying to purify it from this lung cancer cell line. The lung cancer cell line was induced originally from a patient with this hypercalcemia of cancer, of lung cancer. And the protein that it produced, it took us quite some years. It took us about 6 years, but eventually, together with my colleague Richard Whittenhall, who was professor of biochemistry at La Trobe University, and my close working colleagues Jane Mosley and Bruce Kemp, we purified this protein, and Dick Whittenhall determined the sequence, and it was similar to parathyroid hormone, but it differed. It was similar to parathyroid hormone in the first part of it, the first tenth of it, and the rest of it was quite different. But it was produced by this lung cancer, and that was the cause of this hypercalcemia of malignancy. And how it acted was it got generally into the circulation, it worked as a hormone, and it acted generally on the skeleton like parathyroid hormone to promote the activity of osteoclasts and their resorption. And the tumour cells themselves had produced this substance, which we called parathyroid hormone-related protein, or PDHRP. The tumour, when the tumour spread to bone, when it metastasized to bone, it spread in bone by producing this substance locally. So it stimulated local breakdown of bone and allowed the tumour to establish and become established in growth as metastases in bone. So that work had a major effect on thoughts about how is it that tumour cells grow as metastases in bone. Bone is a very hard substance. It's not a friendly location for cells to find themselves into, but when cells get into the bone that produce this substance, PDHRP, they can produce it and resorb bone and allow a niche to be established so that the metastases can grow. And so we worked on that. That was discovered in the mid-1990s, and we worked on it, and we've worked on it ever since. I stopped working a couple of years ago, but we've continued to work on it ever since, in addition to working on how the cells of bone communicate with each other. And this substance was produced by cancers, but we also found that it has local effects in skin. It aids in the differentiation of the cells of skin. It acts on the placenta to promote the movement of calcium across the placenta from the mother to the baby to make the calcium available for bone to mineralize and grow hard. And there are many other effects also in normal tissues of this PTH-related protein that we discovered in cancer. So it's not just a cancer hormone. But that was a major effect. That was a major outcome, a major clinical outcome, if you like. Well, in the very early stage of my work, it was funded by the NH&MRC. I had a fellowship when I came back from London with McIntyre. Had a fellowship to work with Roger Millick for many years, and then I was appointed by the University of Melbourne out at the Austin Hospital and obtained project grants from the NH&MRC and from what was then the Anti-Cancer Council of Victoria. And the Anti-Cancer Councils supported our research with project grants for quite some years, and so too did the NH&MRC. And beginning in 1984, we developed a program grant with the NH&MRC. It's a major grant, 5-year grant that we obtained successively over the next 25 years. So for 25 years, until 2009, I had a program grant from the NHMRC, which helped us to appoint the colleagues that worked with me many times over those years. When I did medicine, I decided that I wanted to know the biochemical mechanisms of disease. What went wrong with biochemistry that caused diseases? And there were plenty of examples that we didn't have any idea what the biochemical mechanisms were. And when I graduated, I still wanted to understand the biochemical mechanisms of disease. And when I was a resident at this hospital, I took a year as the resident of a man named John Owen, who was a Scot, who was in charge of the biochemistry department here at the hospital. And I worked with him, and that gave me the impetus to continue to work in biochemical mechanisms. At that stage, I had also an appointment in the University of Melbourne biochemistry department, teaching biochemistry to medical students, and that inspired me further to want to know, what did all this mean? What did it mean to understand a bit about biochemistry? How did that apply to any diseases? And so it was that which drove me in. And I happened to choose bone because that was what I was exposed to early on, and that's what I found extraordinarily interesting. Well, in the early days, so little was known about bone that once I realized that there were cells of bone, I just wanted to know the truth. How were those cells governed? How did hormones act upon them? That was my starting point. How did hormones act upon the cells of bone? And then as time went on, and I eventually realized that what we did was relevant to, at first, osteoporosis and then cancer, I really wanted to know how the control of these cells of bone related to osteoporosis, and wanted to know, and to cancer. Well, some of my interests outside of work I can't really follow particularly well anymore. I was very interested in fly fishing and got to be reasonably good at it. I played a lot of golf. I can't really play golf anymore. I played cricket. I was interested in cricket, and I'm still very interested in cricket and in golf. I'm interested also in football, and I'm interested in my family and my grandchildren, of which I have a large number. But I don't have any hobbies like woodwork or gardening. I can't stand gardening. So I read a lot. I'm very interested in music, particularly classical music. But I lead the life of an elderly person, and I come into work. When I say work, it's not really work for me anymore. I come in a couple of days of work because I continue to be interested in the ways in which my colleague Natalie Sims continues and develops further interests in how the cells of bone communicate with each other and how they work. And this is an extension of her work that she did with us for many years, but which goes beyond anything that I ever had any direct involvement in. But I find it extremely interesting, and interesting enough for me to keep coming in here while I can.